Hi, everyone, and welcome to the final installment of the Cute Syndrome Foundation CTR series. My name is Shelley Frappier, and I am the executive assistant at the Cute Syndrome Foundation, but most of you know me as Nico's mama. I want to say good morning to those of you on the West Coast, good afternoon to those on the East Coast like myself, and good evening to those that are joining us overseas. I am so excited that we are ending our 2021 CTR series with a session from Neurocrine Biosciences about the very first SCN8A clinical trial. Get your questions all queued up. I know you have lots, and you can ask them over here in the chat. We have a few housekeeping items. Group interaction will be done through the chat on your right. And please ask all of your questions there for our speakers and for Hillary. We'll try to get to them within the session, and hopefully those that we don't get to will get answered, and we'll post those in the support group for you. If you have any tech issues, please type a request for Megan Varner and we will try to get to it and someone will help you. And before we get started, I wanted to thank our platinum level sponsors, Praxis Precision Medicines and Neurocrine Biosciences. Thank you very much. And without further ado, please let me welcome Hillary Savoie, the founder and director of the Cute Syndrome, without whom we may never have seen this trial happen so quickly. Thank you for your tireless advocacy and commitment to SCN8A.
Thank you, Shelley.
You're welcome.
And first of all, before we get started, I gotta say, happy birthday, Shelley. Everybody, it's Shelley's birthday.
Yeah.
And it's also want to wish a happy birthday to her birthday twin, our buddy Max, who is celebrating his birthday in the hospital today and fighting so hard. And we're so proud of him. So happy birthday, Shelley and Max.
Thank you. It looks like there's a big party all set up in his room today. His mom never disappoints.
No.
Oh, wonderful.
Could all be there with him, but I know we're there in spirit.
Yeah. Well.
Okay. So thank you, Shelley.
You're welcome.
And so as Shelley said, this is our final event in our 2021 CTR series. And it works out really well because this series was started 2 years ago to prepare our community for exactly this moment that we find ourselves in. The thing that we're here to discuss with Neurocrine's wonderful team, an SCN8A specific clinical trial. I think as many of you know, I occasionally get a bit emotional talking about this trial, and I'm just going to embrace it because it's there's a lot of reasons for it. The most obvious reason is that it brings a lot of hope to our community, for our children, and for our future. But the other part of this is that I know over years of working first with Xenon and then with Neurocrine, how many people it takes, all of them working and pulling really hard on our behalf to make it to this point. And every time I think about this trial, I can't help but think about JP and Charlie from Xenon standing up at our very first gathering in 2015 in Philadelphia and saying, hey, we think we might have something that'll work for your families. And when I think about that moment, I think about all of the time that came before that moment, all of the work there, and all of the work, all of the days since, including with this team at Neurocrine, who took on this program in 2019 and ran with it in the middle of a pandemic. And I'm so pleased that the team will be joining us today to talk to us more about this program, which we all now know is open for enrollment for the first patient at Cook Children's under Dr. Perry, but also to answer our questions live today as we come together as a community for us, for our children. So with that, I'd like to welcome Brian. I know he's popping back in, Heather, Carolyn, Tyler, to go ahead and turn your cameras on, and hopefully Brian will find his way back in. Is he still having a hard time, Tyler?
Yeah, we suggested he maybe log out and try to log back in.
Okay.
So we're troubleshooting. He's somewhere trying
He's
to
in
get
the ether.
in the ether. We'll hopefully have him soon. Yeah.
Well, please tell him, he's just going to be fashionably late to the party. We all appreciate it. So
Exactly. Hopefully we'll have him, but we're well represented.
well, welcome, guys. I want to just take a moment to say, I know many of the families are familiar with your faces. Carolyn, they saw you in the last CTR series last year. Heather, I think the first they saw you was at the gathering, and Tyler at the community meeting, and Brian in the video that was released in August. So I know you guys are familiar faces, and I've had the pleasure of getting to now meet all of you in person, which was wonderful. So we're old friends, but still, I think it would be nice if we just took a moment to have each of you kind of give a brief introduction, tell us who you are, what you do, and why you do it. Oh, yay. Hey, Brian.
Hi.
You made it.
So glad to make it, just in the nick of time.
Yes. It was a fashionable entrance.
Exactly. I'm
I
just
like it.
fashionably late. And I even lost the tie today. That's my commitment.
It was lost in the ether while you were moving?
Yes, exactly. As I was switching web browsers, it suddenly dissipated.
So we were just going to do some intros. Tyler, I think maybe we can start with you if you want to get us kicked off and then help us make the rounds.
Sure. So my name is Tyler Reagan. I am Director of Patient Engagement and Advocacy for Neurocrine's rare disease programs, of which there's a handful. But I have to say, honestly, I spend most of my time on SCN8A, and that is certainly the one that's most near and dear to my heart. And I've been with the company since March. I've been in the industry for over 15 years, but I have to say, I absolutely love my job. There's so much purpose and meaning to it. And I love the team that I work with. So really happy to be here. Also, before I let the team introduce themselves, I just want to kind of start off with a couple of just additional introductory remarks. First off, I want to thank you for this opportunity, Hillary. And I have to say that this is a much better position to be in. About a month ago, we did our own webinar with the patient community. And I have to say, it's really challenging to try to read questions that have just come in, try to engage in the conversation, try to make sure you're not reading questions that have already been asked or already been answered. So I don't envy your role, but I appreciate you doing that. And also just want to thank, on behalf of my colleagues here, I want to thank the patient community for joining us for this discussion and for your interest in our program. And we are truly humbled by the opportunity to develop a potential therapy for your kids. And we take that responsibility very seriously. We try not to take ourselves too seriously, but we take that very seriously. So thank you. And last, just want to say thanks to Xenon for all their work, really to get us to this point and for their continued partnership. And it was, like you said, it was incredible to meet you in Germany, and also some of the SCN8A families that were there as well. So again, thank you. So with that, I will turn it over. Maybe Heather, if you want to continue the introductions. Oh, I think you're muted.
Unmute.
All right, so we got that part out of the way. Hi, everybody. Thanks, Tyler. Thanks, Hillary. And thank you, everyone who's joined today, for coming together to chat with us. I'm the program team lead for epilepsy at Neurocrine. So I have the job of kind of connecting all the dots and all the things that go on to bring a clinical trial, to bring a therapy forward. I work with our cross-disciplinary teams in regulatory, in chemistry, in the preclinical space, all along the spectrum of activities to put together the plan that moves the compound forward, and we hope eventually to you. So we're super excited about opening the study. We're happy to be here to talk to you about it today. And we are taking a lot of care and putting a lot of work into trying to get it. So nothing is perfect, but what we do need is to have it sufficient for regulatory filing, right? That's what we're looking for. So really important to gather that evidence and have it of a good data quality that we can submit to the FDA for evaluation on the safety and efficacy of this treatment. Why do I do what I do? I can't help it. if you come too close, I'll try and organize it. I'll try and put it all together. So be forewarned. But I really feel so privileged to be able to work on this particular indication. In clinical research, in biotech, in pharmaceuticals, right? There are all kinds of things. There are blockbusters. At one point I worked on gout. So gout is terrible if you have gout. It's really painful, but it's kind of hard to get out of bed in the morning and go, okay, I'm gonna go work on gout. I have no trouble getting out of bed in the morning. In fact, I have trouble sleeping working on this indication, right? It's quite the opposite. But I love that, right? So I love the chance we have to really make a difference with this treatment, with this medication. So I don't know what else there is to say. We are hard at work, and I know that where I'm coming from, our team shares that. Beyond the team that you see on the screen today, and that we're chatting with you, our team, all the way from our CEO to our chief medical officer, down to our finance people, we've shared your stories. We talk about what research we're doing, and I know that our company is really dedicated to this space. So again, thanks for giving us a little bit of your time today. Brian.
Thank you, Heather, and thank you, Hillary, so much for putting this together. My name is Brian Mosley. I'm a medical director of clinical development at Neurocrine. I'm actually one of the newer members to Neurocrine that's on this panel. I joined in March of this year. Previous to joining Neurocrine, I was an adult epilepsy specialist at the University of Cincinnati in Cincinnati, Ohio. Through my training and also as my experiences in attending in Cincinnati, certainly saw the devastation that epilepsy can bring, not only on patients, but on their families, their loved ones, and their caregivers as well. When I learned about the opportunity at Neurocrine, I absolutely jumped at the chance to join the team because I knew that they were working on molecules that would address the unmet needs of folks that I was taking care of in my clinical practice and that my colleagues were taking care of. We certainly have an opportunity here to hopefully make impact on the lives of people who suffer from this disorder. I've just been thrilled to work with the entire team, not only the folks that you see pictured here on your screen, but also other individuals that are behind the scenes. We have such a great collaborative spirit here at Neurocrine. We work not only during the day, but at night and even on weekends as well to bring these trials to light. So I'm just so happy to be able to meet with you today and to hear from you, just like I was able to hear from families in Europe during the SCNA conference that took place last month. And now I will pass it over to Carolyn.
Thanks, Brian. Sorry, I got a little distracted because I wanted to share a fun fact that my brother's name is Brian, so it's wonderful to have a work brother named Brian as well. So we've fallen right into the no sibling rivalry, but fallen right into a great partnership with Brian joining the team. Yes, I can't believe it's been about a year since I first joined and had a conversation with you, Hillary, and some other guests on another WebEx for the community. And it's been quite a year. I'm excited to still be here and in a more official capacity within our clinical development team. I'm a psychologist by training, and I'm working on our pediatric epilepsy programs. I've had a lot of focus as of late on our outcome measures, the non-seizure outcomes, as well as really working very closely with Tyler and Heather to infuse, the patient focus and what that really means in our drug development process. So, one of those things, as you well know, is kind of working on being as transparent as possible, while still, really maintaining the integrity of our trials, because every patient counts and we want to get this right. So super excited that we do have, more information that we can share every day, and looking forward to the conversation today.
Fantastic. Thank you, guys. I appreciate you taking so much time to let us in a little bit on why you do what you do, and what drives you, and what you're passionate about. I think it's really important to see that from our side. And I know, Carolyn, you have some slides to share with us. And before we get into those, I do just want to let the families know we want to take a little bit of a different approach to questions today, just because this is a trial that is enrolling, and we want to make sure that we're protecting everyone's privacy and the validity of the trial, and not exposing any names or things like that. So we've asked Neurocrine to keep their chat function closed. So that does not mean that you shouldn't ask your questions. In fact, it means you should ask your questions and feel very free to do so however you wish, and let me read them, and I will exclude anything that's personal or identifying information as I recount those questions to our team here. And then the second thing I just want to note, again, I know you guys are all familiar with this notion, but when we're dealing with our pharma partners, or when we're dealing with regulatory bodies, sometimes there's questions that they can't answer, and it isn't because they don't want to. It may be because they cannot answer them, or they don't know yet. So we just want to be sure that you guys understand that the intention here today is to have as many questions answered as thoroughly as possible. And I know, having spoken to this team, that that's absolutely the goal here today. So I want to encourage families, as we go along through the presentation, start queuing up your questions, and we will start questions after Carolyn finishes with her presentation. And Carolyn, I don't know if you need help with that's the little computer-looking thing with the X through it, or the line through it.
All
That's
right.
the share screen button. It's not the most obvious share screen button that ever was.
And it goes away when you're in full screen mode. So.
Okay. There you go.
So that works. Okay. And let me get my workspace here. Okay. So, we want to get to the point. We know that everybody has been asking questions about, study startup. What does that mean? Where is it? So here is our latest update. I think since the last time we had the Neurocrine presentation, we've been able to add in cities. As many people may know, we did activate our first trial site in Fort Worth, Texas. And in case people are wondering, I don't think it's a big secret who the likely key opinion leader physician there would be. So Dr. Perry's site is now activated, and we are working to activate further study sites. And you see here the U.S. is where likely those initial sites will open, and we will continue giving updates as they come. And maybe, we can talk a little bit more about what study startup means. I know you might have some questions about that. If you didn't get a chance to see our Neurocrine presentation, Tyler, is that one? Have we been able to share the recording of that?
I am racking my brain here. Hillary, were we able to share the recording?
I don't think we have shared the recording. I think we just had the live version.
Yeah, it was in the review process, which takes kind of an excruciating long time. We have to do internal reviews, as well as we have what's called an institutional review board. It's an external body that reviews anything before it's shared externally. So as I recall it, yeah, it's still sort of winding its way through that process.
Okay. So the kind of Cliff Notes version is, hopefully we can get that approved, but Heather gave a wonderful kind of boiled down version of all the complexities that go into study startup. Needless to say, it's a recipe or orchestra that needs to come together to make music. And there are many kind of aspects of contracting, training, making sure all the materials get to the sites that go into that. And that's why you don't see kind of this whole map light up green with all of the study sites at the same time. So it is kind of a trickle process, but we're super excited to have that site number 1 activated. We think it's great news, and more to come on that as we move forward. So just kind of to give a few highlights as a reminder for the study. The purpose of it is double blind and placebo controlled, and we can talk a little bit more about what that means. But it is primarily to evaluate efficacy and safety and pharmacokinetics, which means what's happening to the drug as it moves through your body, versus placebo or sugar pill as a adjunctive, which means add on to your normal therapy for seizures in individuals who have SCN8A DEE. We are using, in terms of the formulation, a granule form, and I know there's going to be questions about that we will be able to answer. It has been formulated for G-tube compatibility, and it is ketogenic friendly. So what are we looking at in this study? Many trials that are looking at potential treatments for children with a variety of DEEs. Kind of the most common path to an FDA approval is to look at a seizure endpoint. Again, as I mentioned earlier, the non-seizure endpoints are near and dear to my heart, and I know that they're of utmost importance. So we are looking at other things in the trial, but the kind of primary goal is to determine whether or not there is a significant difference between the treatment and the placebo in terms of change from baseline, which is the start of the study, to in the 28-day seizure frequency. And we're looking to enroll 52 participants worldwide, and estimating that it's going to take us until about the end of next year to do that. And so there we go. So while we look forward to potentially opening this to the younger age groups, right now the study is open to people between the ages of 12 and 21. We are requiring clinical and genetically confirmed diagnosis of SCN8A DEE. And in order to qualify for the study, the people with this condition need to have had at least one countable motor seizure per week over the last 20 consecutive days, or the last month. And they have to currently be treated with another anti-seizure medication, at least one, but not more than 4. So we're not asking anyone to come off of their current treatment, even if, we understand that you're coming into this because you don't have adequate seizure control, but we're not asking to stop medications. And then the parent or caregiver is going to be the person that we need to partner with in order to accurately identify the seizure types and complete the seizure diary in this trial. And there is going to be training involved with that. So in terms of this study, there is what we call a screening period. And what does that mean? So, once you determine with your doctor whether or not this is a trial that your child might qualify for, during the screening period, they'll do a variety of tests. They'll try to confirm the diagnosis. You'll be given a diary to keep in terms of making sure that they're having the number of seizures required to enter the study. And kind of it can take up to 8 weeks to gather all of that information together and really be ready to start in the study. So once that's done, they enter the double-blind placebo-controlled period. And that day one is often referred to as randomization or baseline. So randomization means that your child will be assigned to receive either the active treatment or placebo, and that is done in a way that neither the investigator or the family or even Neurocrine knows during the study who is on active treatment. And then this period of treatment will last up to 18 weeks. And following that, the patients who complete the trial can enroll in a long-term extension study where everybody will have the opportunity who enters to receive the active treatment and see and determine whether it's safe for a longer period. So a little bit more about what happens kind of during that double-blind treatment period. There is, you're probably familiar with this, if your child is taking a medication, a titration period. So, they start low and then increase the dose to what we think is going to be the target dose. And that's dependent upon their weight. And then there's what we call a maintenance period. So that's after they reach that target dose, then they're maintained on it for 10 weeks. And at the end of 10 weeks, towards the end of the study, there is a tapered period where they're tapered off of the medication. So why are we what are some of the priorities in this study? So we already talked about looking at that primary endpoint for efficacy to see if there's a change in seizures, but we really want to focus on safety. So as you know, this is the first trial for children with people, because I guess 21-year-olds aren't children unless they're yours. This is the first time that there's any treatment that we're studying in this fashion for this condition. And it's also the first time that this particular compound, our 352 compound, is being tested in these individuals. So safety is a huge priority for us. It's going to be monitored very closely. We're fortunate that we have kind of a whole group of people monitoring that. And of course, Brian, as our medical director, is also part of that team. All medications, as you know, have potential to cause side effects. That's part of the reason also why we have a placebo control, is to really be able to understand, if there are things that your child is experiencing during the study, we really can best understand whether it's due to the introduction of the study medication versus, just something that's happening otherwise in the context of placebo. And then, we ask a lot of questions of families about kind of side effects of their current medications. We'll provide them with as much information as we know of what to expect. And if there is a side effect or what we call an adverse event, that's something that's documented very closely, and it may or may not be a reason to withdraw from the study. But we do have procedures in place for that. All right, I'm going to stop here because what's next, I think, is the immediately what's next is that we want to enroll our first patient. So I think now is a great time. I'm going to stop sharing my screen to go to our Q&A, and I can bring back up the slides if we need to at any point.
Okay, thank you so much, Carolyn. And I know we are, Tyler is correct that we're going to make that slide deck available to share with the families. And I think there are a few more slides with some basic background information. And I think, in discussions with Tyler, we really wanted to make sure that we gave families time to ask their questions and then we can share that resource in the support group. And if you have further questions, as Tyler said, he spends all of his time, majority of his time on 8A right now, and that's because I just keep sending him questions. So he opened the door. So with that, I think we can dig into. I had some questions preset, but it seems like what I'm seeing in the queue is very similar to the questions that I had preset. So I'm gonna try to scroll through these and hit some of the themes. Out of the gate, I mean, unsurprisingly, the biggest set of questions that are popping up are when will various sites come online? So just to hit a few points, and then I'll let you guys answer. Anticipating, is there anticipation, and if so, when, for a site in Canada? How many sites in Europe have already been selected, and when do you plan on enrolling first patients in Europe? I had a question ahead of time that was very similar, specific to UK. What about sites in the UK, since they may have some different rules than the rest of Europe? And then a particular question about sites near Philadelphia. So I'll let you guys start with that.
So, okay, I'm gonna reshare this. So this gives you a sense worldwide of what we're looking at in terms of potential study sites. I wish I could give you like dates of, okay, this site opening on this date, that date. Unfortunately, we don't have that information. As I alluded to earlier, we're working to get all of these sites up and running as quickly as possible. In the US, there's kind of your common, I don't want to call them challenges, but just steps that we need to take. Contracting, making sure that, all of the various study materials are on site and the sites are activated, everybody's trained on the protocol. IRB approvals. So every institution, you mentioned Philadelphia, for example, Children's Hospital, their internal review board might meet on a different kind of schedule, and has to go through all of those steps. So as soon as they're kind of getting close, then they set an activation date, and we'll have a better sense. In Europe, it's a kind of a separate timeline, as they have to do separate country submissions. So more to come, but these are just to kind of give you a sense of which countries, including Canada, that we've selected potential study sites, and we aim to get them opened over the next several months.
Okay, so I noticed that the UK is not listed, and is that correct? Currently, is not on the.
I do, yeah, it's not there.
Okay.
I don't know if Heather or Brian have anything to add to that.
I would say with regards to the UK as well as other European countries, we're certainly exploring the availability of sites, the interest of sites, the ability of sites in those countries to conduct this study. And so we're certainly conducting review of sites in the UK as well as other countries that may not appear here. And things may just be at a different stage as well. So certainly, as you know, we're further along with sites in the United States now that we fortunately have one that's up and running in Texas. But that doesn't mean just because you don't see it on this list that eventually during this study that it won't come into play. And so just be aware that we certainly are looking into additional sites. And certainly you can always check clinicaltrials.gov to get the most updated list of sites that are active. And that includes not just in the United States, but that includes in other portions of the world as well.
And as I think the families here should know, as you have information that you can share and can be shared publicly, you have been letting me know, just like with the Fort Worth site, so that then I can pass that information along. My team can pass that information along in our support group and through other means as it's appropriate. So
yeah. I would just say on clinicaltrials.gov, It's not actually real time because we can only update it after we've activated it. Anyway, like with everything else, there's a whole process behind the scenes of approvals and things we have to work through. But it is true that when we do open a site, we will be in contact with you. And also on clinicaltrials.gov, what it'll say is city. So I think we're in the process of adding Perry's, but it'll say Fort Worth clinical site in Fort Worth, Texas. So there is a contact email there that you can reach out to someone at Neurocrine for more information about it. And we'll also be in touch with the community via the advocacy.
Thank you.
Sorry, I just had one thing quickly. I know that even before I started at Neurocrine, that Neurocrine reached out to you, Hillary, to sort of identify potential sites around the world.
Yes.
So there has been behind the scenes. There has been for a long time kind of behind the scenes work going on to try to identify sites where patients are located, families are located, KOLs are located, in close collaboration with Hillary.
Yeah, and for the family's background, Neurocrine was really fantastic about this. With Shelley's help, we were able to map everyone that we know in the world, and with then the help of our partner organizations across Europe and Brazil, to map improve our numbers for understanding those areas, because we don't have full visibility that Cinzia, for instance, would have in Italy. And then feed all of that information back to Neurocrine to help them think about, known populations that will be easy to be in touch with, that are already talking to each other. And then likewise, just from a site selection standpoint, we have had some families reach out saying that their doctors are talking to them already about this trial, even though it's not an active site. So that is another way families can be getting up-to-date information, is to talk to your clinician, especially if they're in a major institution, and see whether this is something that they know about yet. And most likely your doctors, if they are, will be talking to you if they think you fit the criteria. So another kind of theme here, as you guys probably are not surprised to hear, is age range. So for those who have, as Carolyn said, I think your child is still your child if they're 21, but when you would be opening to children or our children over the age of 21, and when you anticipate having the 2 to 11 age group open specifically in the US.
So it sounds like there's two different questions there. One is, will there be a trial available for individuals over the age of 21? And then, also the big question of what about the under 12s? Is that what I'm hearing? So kind of the either end of the spectrum.
Yes.
So it is our hope that, as soon as we have some of that additional data requested by the FDA, that we will be able to extend this current trial down to the age of 2, I believe. I don't know, not to put you on the spot, Brian or Heather, if you want to share anything about people over the age of 21.
We're on the spot anyway. It doesn't matter. I'm going to set it up for Brian. So the age range is tricky, right? Because this is the first trial in SCN8A. We could even, think about, we know the age of onset is on average 4 months. So why not neonates? So if I start there, there is a whole set of requirements to get into neonates. There are formulation requirements, preclinical testing requirements, etc. We don't have that data package yet to address that. We're absolutely interested in it. But really, taking on this compound from Xenon, we are working to get it to you as quickly as possible. Again, kind of like I said at the beginning, it's not perfect. Right? There are other things we could do and we'll plan to do, but we're taking what we got, we're optimizing it as much as we can now, and trying to get it out into to patients, because we still don't know if it's going to be effective. We would like to open the trial to 2 to 11. As you know, from some of our public comments, we have been asked by the FDA to generate some additional preclinical data to confirm the safety of the doses that we've selected in those younger children, which is why we're starting at 12. Those studies are ongoing. And until we have the readouts from those and discuss them with the FDA, we'll still be at 12 and above. We will open the study as quickly as possible, and we're hoping it won't be too far into next year that we'll be able to do that. But again, remember, this is research and it's experimental. And so we don't know what the results of the experiment are going to be yet. So we still have to have the new data.
And to clarify that, Heather, to looking at that 2 to 11 age range, it would be the same trial, just opening a new population. It's not a separate trial. So it's
That's right.
all of the infrastructure is in place. It just
That's
means that Dr. Perry, for instance, can then look at that other patient population.
Absolutely.
So it can happen quickly.
Yeah, so right, it's all of the same, the same schedule, the same visits, the same trial duration. The doses are already formulated by weight to accommodate, down to the younger ages. So yeah, so it's set up to enroll the 2 to 11s. Yeah, and then as far as older than 21, I mean, I think we're kind of pushing the envelope already at 21, to understand, what the course of SCN8A looks like through the disease, and through, how long you've lived with SCN8A and the seizures, etc. And that's where I go, hey, Brian, do you want to say some more about that?
Thanks, Heather. I appreciate that. I would say certainly we understand that people across the age range, both, as Heather mentioned, below the age of 2 down into neonates, but also above the age of 21, can suffer from this devastating disorder and the terrible seizures, the developmental delay, and many other health conditions that can go along with it. And we certainly, as part of our goal at Neurocrine, we want to be able to help all of you if it's possible. For right now, because we know the biggest burden of this disease is in the age range of 2 to 21, we have chosen to focus our efforts in getting regulatory approval for getting a trial launched in this age range. And as Heather mentioned, we are going to start with the 12 to 21, and then pending the availability of some additional data, which is still pending, we'll then go to regulatory agencies to hopefully be able to bring it to the 2 to the 11 age range very soon. But we wanted to focus particularly on the 2 to 21 age group initially. That isn't to say that this drug will not be eventually something that we study in different age groups. But for right now, our biggest focus is to be able to bring this trial now currently to children from the age of 12 up to even adults to the age of 21, and then hopefully eventually down to the ages of 2 to the 11, pending this additional data.
And correct me if I'm wrong, but assuming we would get through to approval, which I know there's a long path there still, there's no reason that this drug couldn't then be prescribed sort of off-label for a slightly different patient population, meaning, over 21 for an adult. Is that correct?
Oh, Hillary, we can't talk about off-label use of medication.
Oh, really?
We would never recommend that.
Well, okay, so
I will add, this is not specific to SCN8A, but what you may be alluding to with some of the questions is, we have indicated on our website and publicly disclosed that we are pursuing trials in focal onset seizures in adults with the same compound.
Okay.
So, focal onset seizures, we will be looking at that specific as a seizure type, but not,
Okay.
tied to SCN8A.
Okay. So a couple of other questions I think that are coming up just about clarifying the patient population. There's a question about, and I'm gonna push these two together because I think they have similar answers. If a variant has an uncertain effect, a gain of function or loss of function, will there be functional study prior to enrollment, or when someone is given an SCN8A variant, meaning a genetic test result, is it automatically a DEE? So I'm going to push those two together because I think that helps you guys describe the difference between the sort of genetic result and the clinical requirements that are required.
So this became a hot topic while we were at the meeting in Germany, because I know there was some discussion around that. As part of this study, there is kind of an independent diagnosis confirming panel on which, we do have geneticists in that panel to really look at the specific variants, because our hypothesis is that, having a NaV 1.6 sodium channel inhibitor may help somebody who has a gain of function mutation. And we have, I think there's still a lot to be learned. And I understand that not every variant has undergone that functional characterization. So it's certainly something that we're kind of internally discussing after all of that hot topic came up in Germany.
And then I think also for the, if someone has an SCN8A documented variant that's of, that is considered likely pathogenic, or a VUS that's suspicious, or a known pathogenic variant, I think the answer to the question here about that's not automatically a DEE, because we have individuals who have known variants who do not have epilepsy phenotype. Mm-hmm. So, and may have a movement disorder kind of a phenotype or another phenotype. So to answer that question, I think that's sort of dealt with in your inclusion and exclusion criteria. Right. The individual would need to be having seizures on a regular basis in order to qualify.
Correct.
This is a great question here from someone who always asks great questions, but who will remain nameless today. So he asks, regardless of initial outcome on a 12 to 21-year-olds in the study, depending on how long it takes for FDA to approve data for younger patients, will you try to test younger patients, considering the cases of, and I'm going to massacre the name of this drug, I always do, as gabapine and retrogabapine for Millichap's, he references Millichap's 2016 study, where the effectiveness was only seen below a certain age in KCNQ2.
And so I can go ahead and address that. And absolutely, we definitely, provided we have the data to support it and that regulatory agencies allow us to do so, have every intention of studying this medication in children down to the age of 2 years. And so however long it takes us to recruit children above that isn't going to stop us. If it was just suddenly all of our trial filled up with 12 to 21 years olds, it's not going to stop us from exploring that age range.
Okay. And another great question, considering this medication has passed phase 1 trials, are there any plans to apply for compassionate use, considering certain variants are well characterized in vitro with multiple mechanisms of failure? He gives examples of 850 and 1872, and are highly unlikely to have seizures be fully controlled.
So at this point, we still don't have evidence of efficacy for this drug and safety in this population. And that's really what this trial is about. Okay. So I think it's likely premature for us to offer compassionate use. We are aware that there will be patients who don't qualify for the inclusion-exclusion criteria. And and we're thinking about how to address that. But at the current moment, we don't feel that we have enough data in the patient population to make sure that it's safe. And so that is really the point of this trial.
Okay. We have a couple of, let's see, there's some that we've kind of answered to a certain extent, so I just want to be sure. Okay, question. The plan is to enroll just 52 patients or at least 52 patients?
I can answer that one. So it's 52 patients, and there is a little bit of buffer in the protocol to enroll an additional 8 patients should we need to. So our so we have a little room there, and that 52 patients is 2 to 21. So it's everybody. It's yeah.
Okay. So that's important, right? It's, again, looking at that patient population that's enrolling currently, which we hope will extend to the 2 to 20, the 2 to 11 to include those. That number, that larger number, doesn't change. We're still looking at 52 for that full.
That's right. That's right.
Okay. Also questions about open label extension. So, if my child receives the placebo and is in the trial, will they be able to access the compound when the trial is complete? And I think a sort of follow-up to that is, you mentioned the taper period. Presumably if there is an open label extension, does that taper period then not happen and you just extend into an open label extension?
So I can go ahead and address that. So absolutely, there will definitely be an open label extension. We use the term active extension here internally to describe it, because to hit upon the second point, there actually will not be a taper. So some studies, when you go into an open label extension, you actually come off drug or at least go to a lower dose before you then enter into the open label extension. With this trial, depending on whether your child who is in the trial is on study drug or placebo, will enter into that extension trial on the same dose, and then in a blinded fashion, meaning you won't know, the investigator and the doctor that's treating you won't know, and the rest of the site staff won't know. If your child happens to be on placebo, will then be converted over a number of weeks to actual active study drug. And so this is a unique circumstance where we wanted to avoid having to do a taper. We know that the seizures in SCN8A can be not only quite severe, but quite frequent as well, and didn't feel that a taper would be in the best interest. And so that's why we designed our open label extension, or what we call our active extension, in this way.
Okay, so sorry, this is actually, I don't know the answer to this question, so I'm asking for my own education. So basically, you would enter into the open label extension not yet knowing whether you had been on drug or on placebo.
Correct. And you still won't. We will be maintaining the blind throughout that trial. But you will know that as soon as you are outside of what we call our blinded dose conversion period, that your child will be on study drug.
Okay. Oh, that's fascinating. I did not know that. So I just want to repeat that for the families in case that wasn't clear. So basically, if you start the trial, you have your monitoring period, then you're either in a placebo group or the active group. And if you are reaching the end of the trial and you want to discontinue use for whatever reason, you would have a taper period and you would discontinue. But if you were entering into the open label extension and you were on placebo, you would be slowly increased, not knowing whether you had been on placebo or not, until you were on the full dose of the drug in the open label. And then if you had been on the active drug, you would just continue on the same dose. And everyone, once they're in that full period of the open label, will be on full drug dose, but you still will not know if you had been on placebo before.
Correct.
Okay. Fantastic. Thank you so much for explaining that. CBD oil, is it counted as an anti-seizure medication?
So certainly when it comes to the trial, we realize that a lot of children with SCN8A may be on Epidiolex. And so that definitely is allowed as one of the what we call concomitant or other seizure medications that you're on. We're expecting that children will be on at least one, if not up to 4 anti-seizure medicines that are allowed that will remain unchanged during the trial, and Epidiolex is one of those. Now, for safety concerns, because we want to make sure that during this trial, not only are we looking at the safety of the drug that we're studying, our unique sodium channel blocker, but also how it interacts with other medications, side effects that may result when it's used along with other medications. We want to make sure that the medications that are used concurrently are those that have gone through the same rigorous approval process that our drug that we're studying is going to go through right now. And so because of that, when it comes to medicinal marijuana compounds, we have chosen to only use those like Epidiolex that have received FDA approval. And so certainly, as I mentioned, if your child is on an FDA-approved medicine like Epidiolex, they certainly can potentially be in this trial as long as they meet all other inclusion or exclusion criteria. But as far as other medicinal marijuana compounds are concerned, that would not be a definite, just because we once again want to very rigorously look at the safety and the tolerability of this medication, and particularly as it relates not only to the drug itself, but how it may interact with other medicines that are on the market.
Okay.
Yeah. In the CBD space, I realize there are some that are more maybe rigorously prepared than others, but I think the main issue for us is that introduces a lot of variability, and variability hurts our data integrity, right? And makes it harder to interpret the effect and separate the effect of 352 from anything else. And if we have that kind of noise in our study data, that will give us, that will be a problem for us to determine how efficacious 352 itself is.
And again, I think for the families, it's a really important distinction between understanding that there's the study, right, which is an experiment, and the goal is to make that data as clear as possible with the ultimate goal of approval, so that then the drug is available to everyone, even those who may not be seizing as frequently, or those who might not fit the strict criteria of the study, but or in its strictest form, but would still be good candidates for the drug being used.
In case it wasn't obvious, on our company jargon, 352 is our name for the investigational drug. So probably context, you understood that, but just to be sure.
Yes, which we all call 901.
Yeah, 901, Neurocrine 352. We all have our
and anxiously awaiting a fun generic name that will be introduced at some point
that will roll off of our tongues after like 3 days, and no one else will be able to say it. So actually, speaking of that, I know we're close to time. I'm tending to want to let us go over a little bit, but I don't know if you guys have to jump off for another call immediately. But if you have time to hang out a few minutes longer, there's just a few more questions I want to be sure that we wrap up. Another great question from, let's see. So are there concerns with patients that may have variants close to the proposed binding site for 352, 901 study drug, for example, 1617? In response to the medication?
So I would say with regards to mutations themselves, one of the things we want to make sure is that it is a gain of function mutation. And when it comes to, as Carolyn mentioned, what our diagnosis confirmation panel is looking at, what they're focusing on with the biggest intent is whether it's a gain of function, because there are children who have SCN8A mutations that are loss of function, and we don't believe from the preclinical work that we've done that those children would benefit the same way from a seizure perspective from using this particular medicine. And so that really is our biggest concern on the genetic front. And so that's why we have a panel of experts, including a geneticist, who will review that information. And certainly if there are concerns about the location of the mutation, we would trust that particularly the geneticist that is on this panel would bring that to our attention. And if there was any thought that your child, if that happened to be your child's mutation, wouldn't potentially benefit from this mechanism of action, a reason why your child would not be somebody who we would really want to put in this trial, not because we don't want to provide help, but we want to make sure to not expose your child to a drug which wouldn't potentially provide help and may only lead to the development of side effects.
And I think,
sorry, I was just going to say as well that there's no particular specific gain of function mutations are excluded. We just don't know enough at this time to exclude any particular mutation once we sort of determine that the phenotype is gain of function. However, if we do know the exact location, that will be something that we look at in terms of response to drug, right? That's something that we can look at in our analyses, but it's not excluded upfront.
My understanding, to do my best to answer from my background to this individual, is that they are aware of the potential issue for binding site with 1617, but that because there's a wild type as well as the mutant form, that it should still bind on the wild type and thus knock down the flow of sodium through 1.6 on the wild type, which should even out the expression ultimately. So I think there was scientific thinking behind the choice not to exclude any mutations in that particular location, because that is a very common, relatively common mutation, which is a gain of function, and potentially, theoretically could be slightly problematic. But I don't think that the thinking is that it should still work.
Yeah,
but that's me speaking as me.
I'd like to add just a couple of things that come up with me for this. So one of them is, back to, the slide I shared about monitoring for safety. We are rigorously watching the children that are enrolled, the people enrolled in the trial. And certainly if there is any adverse event that happens after being enrolled in the trial, these will be very closely looked at. And of course, there are certain criteria where the clinician, the study doctor would say, okay, we think we need to take you out of the trial. Because of course, utmost concern is safety. But the other thing is, if after talking with your doctor, or even after going through the screening process, for some reason, the patient does not qualify, then, I think, Hillary, we talked a little bit about all of the different ways that the community can contribute and support. And, in this discussion, one of them is encourage others to get genetic testing, and you participate in efforts to share your genetic results and get the functional characterizations so that the field itself can really increase our understanding of the kind of genotype phenotype correlations and who really is going to benefit from different interventions.
Fantastic. And then we have a question that's sort of location specific, but I'm going to generalize it a little bit, and then a few follow-up questions for some of the things regarding some of the sites that are in countries that you've indicated there will be studies in them, but they are not up and running yet. We have a family that's indicated that their doctors are making them aware in that country that there's going to be a study. Are there things within that country that they can be doing as advocates and parents to help coordinate and lay the groundwork for easing that trial off the ground?
I'll wade into this. I know we're still, as a team, learning and understanding the regulations in individual countries. Certainly, if their doctors are kind of alerting them that this is coming, it's really talking with them directly about what they can do to help. And like I said, kind of doing what you do in your community and spreading awareness and about SCN8A, what it is, and maybe really encouraging the identification in people who don't have it. But yeah, work with your doctor in those respective countries to stay up to date on kind of when it's happening. And again, of course, we'll let you know as soon as we can when these sites are opening.
Yeah.
And to repeat the 2 to 11 age range, as you expand, the full number of the study participants will not necessarily increase by anything more than another 8. But the hope is to open that age range in all of the various locations that you have studies
Yeah.
eventually, once the overseeing bodies have approved.
That's right. It'll be a global protocol amendment.
Okay.
And in fact, in the protocol as it exists now, it is clear about the intention to open to the younger age range. So,
and is that the same for FDA and EMA? Meaning, once FDA approves that expansion, do you predict that EMA also will, or do they have separate requirements from you?
they have separate regulatory requirements. So we'll file, it's a protocol amendment, and we will file, as when I say a global protocol amendment, that means we're going to file it in all countries.
Okay.
So I can't see the future, but as I said, we did explain in the current protocol that we would be intending to amend and open the age range. So with, when the current protocol is approved, that's sort of an implicit, you assume that they read and understood the intention, and that would ease the way for opening the age range. But again, it still has to go through all of the steps and approvals.
Okay. Two more, I think, clarifying questions. The open label study or the active study, extension study, does that, how long does that extend for?
So currently, as it's written, it would extend for up to 2 years. And so that is what is written. Certainly, depending on how things go, that may change. But as it is written right now, it would be an additional 2 years of treatment.
Okay. And to clarify on CBD oil, does that mean what you said, Heather? Does that mean that the unregistered CBD oil, hemp oil, will automatically exclude trial participation?
That is an exclusion criteria for us. So,
okay.
Yeah.
And I guess just to clarify,
don't do anything right now. This is the thing I'll say. Please don't take matters into your own hands and try to optimize yourself for the trial, your children for the trial, right? Really, this is going to be a matter of talking to the site investigators and determining the plan for evaluating eligibility. And if they're on something like CBD oil, talk to your doctor before making any changes.
Okay. And then just a clarifying question about the 1617 variant, confirmation that yes, it is a gain of function and it is within 5 amino acids of the proposed binding site. And that was the source of the question. We have some scientists amongst our parents, so and we love it. We absolutely love it. So on that note, I know we've held you over. This is the first time I think we've gone this far over time, but I felt like this was the one to do it on. I really wanted to be sure our parents had the chance to take full advantage of being able to talk with you. I don't think I asked a single one of the questions that I had prepped, although I think the families actually basically got them all set for us. I just think this is, first of all, I just want to thank you guys for coming on here and being so open and willing to share. And, this is a space that we're all learning together. And I think it's so important to the extent that we can to put this on display for our families and let them see this process and know that you guys are here to answer their questions. And we're just also grateful for your time and for your dedication to our community. So thank you for coming. And again, this is just the perfect note to end this series on. It was nearly 6 years ago today that we learned about what was then 901. And here we are at the end of our second clinical trial readiness series and talking about the details of actually enrolling our patients. And again, thank you for coming here to talk so openly with us. And thanks again to the TCSF team, Casey, Megan, Shelley, who make this look easy, and our event sponsors for this series, Praxis and Neurocrine. And I would say, see you guys all again next week, because that's what I've been saying for 4 weeks or 5 weeks. But I won't see you next week. I will see some of you in our moth workshop coming up. We still have, I think, 1 or 2 slots open. So if anyone is wanting to sign up, do it now before we finalize everything. Let me know or Shelly know if you're wanting to sign up and can't find the link. And then see you in book club, I guess. And when we will see all of these folks and at the gathering again in December. So thank you guys. Have a
Thank
wonderful
you.
week.
Thank you, Hillary. Thank you.
Thank you.
To Thank you.
Take care, everyone.
Thank you.
Thanks all.